the late time we start. We continue our session with Julia Yeomans, active nematics and active turbulence.
Okay, thank you. Right, morning everyone. So in the first lecture, I talked about an introduction to liquid crystals. And then in the last lecture, we looked at the basis of active turbulence. And I hope I managed to explain to you where active turbulence comes from, the physics behind it, and also the idea of motile topological defects. What I'm going to do today is, first of all, talk about a small research problem to give you some idea of an example where one finds topological defects in real systems. And this counts as cute example. It's not a sort of generic thing that everybody needs to know about. And then I'm going to talk about what I've called advanced topics. It's not so much that they're advanced as that they sort of don't quite fit together anywhere. Because we want to think about 3 dimensions, obviously. And I rather disagree with Francesco that you don't get active matter in 3 dimensions. Although with active colloids, he's dead right, okay? So we're going to talk about the difference between 2-dimensional active matter and 3-dimensional active matter. Then I'm going to introduce something called active anchoring. Active anchoring. And what this is that if you have a surface of an active nematic, well, if you have a surface of an active nematic, the order parameter is going to change over the surface because inside it's an active matter and outside it's, say, an isotropic fluid. Changes in the order parameter give you stresses, give you flows, and we're going to end up with something called active anchoring. And I'm going to give 2 examples, research examples again, about where we actually see this active anchoring. Then I'm going to talk about what happens if you take these active nematics and put them in confinement. And then the last thing, if I have time, is I'm going to talk about other instabilities. Remember, the one we had yesterday was the fact that these systems become unstable to activity. A nematic state is not stable to activity. But that's only the beginning of it. I know at least 2 other instabilities that we might have to worry about. I've made the list there because really it is, I'm afraid it's a bit of a sort of bitty lecture. It doesn't all fit together. We're doing one topic and then the next, and then the next, and then the next, but all based on the sort of outline that I talked about yesterday. Now, for my sins, I have to talk this afternoon as well. Probably even worse for you if you actually have to listen this afternoon as well. But so what I've tried to do is put the examples in this lecture being different from the examples I'm going to talk about this afternoon. And what I probably would have done at the beginning of this lecture is do a summary of what we did yesterday. But given that I'm going to have a different audience this afternoon, I really need to introduce active nematics before I get started. So if you're coming this afternoon to this colloquium, the first 20 minutes, it'll be great because you'll know what I'm talking about. You'll have heard it before. It will be a summary of what we've done so far. And then I'm going to do 3 examples there, which I'll do in less detail than I normally would in a lecture rather than a colloquium, but those will be new. But you'll have a head start on the rest of the audience. So that's what we're going to try and do. That's what I've just said. Remember that if you're not going to remember anything else. And we'll talk about the hare and the tortoise story. You know about the hare and the tortoise? Do you have them in Brazil? Yeah, okay. My collaborators wouldn't let me title the paper The Hare and the Tortoise Story, but I should have been. So this is actually topological defects in a bacterial colony, and they have a strange effect. And of course, because this is real research, these are the people I collaborated with, and in particular Armin, who was at the time a postdoc in Oxford and is now a professor in Copenhagen. So these are crawling bacteria, 2-dimensional layer of bacteria, dense bacteria, they really are a very nice nematic because, I mean, they're just nematic shapes, and you can certainly see the short-range nematic order going on here. And you can also see something which looks pretty much like active turbulence. It would be even more turbulent if this movie would go a bit faster, but this thing is again active turbulence. And if you plot things like the velocity correlation function, it looks the same as we had yesterday. Yeah?
To the previous lecture, Francesco, why don't we get here global polar order?
They're not polar, they're nematic.
Okay.
They're nematic. Cool. And in fact, it's subtle, right? Because these things have a nematic shape, and then they can have polar order, and they can have polar motion, and they can also have nematic motion. These things actually have a nematic motion. They go like this. They twitch like this. Okay, so what our colleagues in zoology wanted to find is what happens if you mix two different sorts of bacteria here. If you have two different sorts of bacteria which move with different speeds, what happens? Because the biologists are really interested in colonies of lots of different sorts of bacteria, and lots is difficult, so let's start with two. So what they did is they had wild-type bacteria, i.e. ones they got straight from the pond, and then they got genetically modified bacteria which had more of these pili around the edge. These pili are little hairs, and they use them to move. So these things pull on the surface to move. So we've got a mixture of bacteria with the normal number of pili and ones which have more, and so these guys here are about twice as fast. Big distributions, but the average here is about twice as fast. This shows the PDF, the probability distribution of the cell speed, and the brown ones are these hyperpileated things, so on the whole they have a larger speed, and the blue ones are the normal wild-type bacteria. For a long time, I thought wild-type meant fierce, but it means straight from the pond. Okay, so now let's see what happens if we allow these colonies to expand. They're expanding because they're moving into space. On the whole, these things don't divide very quickly, so we can forget about cell division. But if you've got a load of them here, what's going to happen is they're going to tend to expand in this direction. These are the slow ones, and these are the fast ones. And don't worry about those. These are just ones who don't do much at all because they've taken all the feelers off. Okay, so let's see what happens as a function of time. So these are the slow ones. Okay? But then they suddenly overtake. Let's play it again just to see that. Okay, these start off slow, which is how they're meant to be, but then hare and tortoise story, these guys, which are meant to be slower, really quite seriously overtake the other ones. That was surprising. Why? So let's look at the front of the colony. This is the front now of a mixed colony. So we've mixed together the fast bacteria and the slow bacteria. At the front of the colony, you just see the most energetic ones getting there. But then at later times, we can look and see what happens at the front of the colony. The fast ones are yellow. They've been stained yellow. The slow ones have been stained blue. So what's happening is at the front of the colony, it starts off yellow, fast ones, which is what you'd expect. But then at later times, the slow ones are dominating the front of that spreading colony. What is going on? These were in there, so I left them in there. Okay, so because it's this lecture, it's going to have something to do with topological defects. We can identify topological defects in this bacterial layer, and these show the pluser halves, which here are red, and the minuser halves, which here are blue. They're not very long-lived topological defects, but you can identify them. And strictly, they're not topological, because a real topological defect, you need long-range order. And here we've only got short-range order, but people always talk of them as topological defects because they have the right director structure around themselves. At the time, we wanted to model this. We've seen this one before, right? This is the continuum model. We know what happens there. We talked about that yesterday. Another way to model it is in terms of rods. This would perhaps be the most obvious way to model that, right? So we have a load of rods. In fact, we have polar driving on these rods, and we have excluded volume interactions between them. So just a very simple setup here, hard rods, each rod subject to a constant driving force. And what you end up with, again, is a very similar system where you can identify topological defects. I don't entirely understand why we end up with exactly the same story in this hard rod system as you do in the continuum system, particularly when we have polar driving there. Other people who've worked more on these rods might be able to say something better, so I'm sorry about that, but I still think there are questions about when you take this limit of looking at a microscopic system, which in this case is, in theory, dry because we're not putting in any fluid and hydrodynamics, quite why we get the same answer. But I'm going to pass on that, but happy to discuss it with people afterwards. I've said we get the same answer, but why did I say we get the same answer? Well, to compare those three different things, to compare the experiments, the continuum simulations, the hydrodynamic simulations, and the rod simulations, we plotted the velocity field around these defects. This is the velocity field around a pluser half defect. This is the velocity field around a minuser half defect. You can see they look the same. This is the continuum simulations, and there are no fluctuations, so it's really easy to measure these velocity fields. I said the pluser half defects are motile, so this one's busy moving off in that direction. Here you can see the threefold symmetry of the minuser half defect. If you look at this rod model of rods moving around, now you get more fluctuations, but you can see you really do get the same pattern of flows of how these rods are moving on average around the defects. This one here is the experiments. Really horrible fluctuations, but you still see the same thing. You see the same patterns of these two vortices around the pluser half defect, and round here you see these 6 sort of rolls of vortices around the minuser half defects. So that's why I'm saying that the 3 things are the same. It's possible if we measured other things we wouldn't see this, but the velocity fields around the defects are the same-ish, near enough. Okay, anyone got any questions so far, yeah?
Microswimmers are pushers, or?
No, we're just using a polar force. We're not really even having a swimmer model. These are just rods pushed by a polar force.
Okay.
So they're just pushed in one direction. They're not swimming.
Mm-hmm.
Yeah.
Thank you.
And this polar force acts in the center of mass over the direction of the polar axis or or?
And the direction of the polar axis.
Oh, okay.
Yeah. Yeah. Okay. So, oh, is that, yeah, all right. I like these. We have the Easter Bunny in the UK. I don't know if you have the Easter Rabbit as well. Whoops. All right, so now let's look at, this is an experiment actually looking at this mixture of fast bacteria and slow bacteria. This is what happens as a function of time. You get these clusters forming, and this is the same thing again. You get these clusters forming. And it turns out that these are clusters of fast bacteria. So let me tell you what's happening and then give you the evidence, because it's probably easier that way around. So what actually happens in these systems is that 2 plus a half defects sort of come towards each other. They're repulsive, so it's a bit like planets. Well, no, not planets, that's right, wrong. So they go round each other like this, 2 things like this, like charges, move around each other. And so they set up flows because they're moving. And these flows tend to push the bacteria, so they're pointing out of the plane. Bacteria are moving in the plane. 2 plus a half defects come on. What happens is that these things end up pointing upwards. Then they get stuck. And then more of the fast bacteria come and get stuck as well. And the fast bacteria get stuck in these verticalized clusters. You need evidence to believe that. So I'll show you the simulation evidence first, and then I'll show you the experiments, because you can do prettier pictures with the simulations, but the experiments are very clever. All right? So here are a couple of plus a half defects coming towards each other in a rod model. And what you can see is that the rods point up and point towards you and get stuck. Here, the defects were moving slowly to represent the slow bacteria. You get this pushing up, but there isn't enough energy to keep them there, so they fall back down again. The fast bacteria, the rods are pushed up, so they're verticalized, and then they get stuck. There's enough energy to stick them. I'll just do that one again. One on the left is slow. One on the right is fast. I think I now have a movie of lots of these things. Okay, so you can see for slow bacteria, this is a simulation. Nothing much happens. For the faster ones, you get these regions formed of things being verticalized, in inverted commas, pointing, it's a nasty American word, all right? Things pointing up like that, which are stuck. And those clusters you were seeing in the experiments are where the bacteria is stuck. These are the same thing in experiments. I wouldn't have believed the simulations without the experiments. We can make the simulations work, but to some extent we're cheating. We're putting in parameters we want to put in. So these are the experiments. Here are the plus a half defects. They come towards each other, and this region here is where the bacteria are ending up pointing towards you. Let's play that as a movie. These are really hard experiments. Ollie Meacock is the person who did them, because you have to find the right place to look at, and it's pretty small, right? So this is the verticalized region. This is what happens if you color the things, and you can see that the stuck bacteria are the yellow ones, the fast ones, and looking side on, here's the layer, and here are the stuck bacteria. Yep.
Just a technical question. You're really seeing from the side, or is a confocal microscope?
This one is from the top, and this one is from the top. So these are pointing up towards you, and the ones round here are flat. Yeah. And then if you look from the side, all right, You need to rotate that way to get the same. Here's the flat layer, but here are the verticalized ones. So this is a line across there, seen from the side.
What I asking if you really in the experiment were looking from the side, or is a confocal?
It was a confocal
Okay,
looking from the
confocal,
top.
yeah.
Yes, sorry, yes.
Yes.
I have a question too. So you mentioned before that those topological defects are short-lived, right? And
Yes.
short-lived, right?
Fairly short-lived, yes.
And what's the main mechanism for the vortex or the topological disclination or so disappearing? Is that the escape for the third dimension?
No, this is
that
plus and minus meeting each other. So it's the
that's
same
okay,
reason.
but that's usual, right?
Yeah, but there are a lot of them. So what happens is that they do meet each other very quickly.
Because of activity, they
Yeah.
move more and okay.
Yeah,
And they
because
can
there are more
explore.
of them, and so they hit each other more, and it's probably a bit easier to make them.
Okay, they don't disappear alone. They cannot.
Yeah, no, they
Okay,
don't appear alone.
Okay.
Yeah. Good. Other questions? Because this is sort of the
yes.
conclusion of this bit. Yeah.
The experiment is they adhere to the substrate.
They
and so there
move
is-
on the substrate,
Okay.
yes, and they are held down to the
There is
substrate.
no confining plate above.
No,
Okay.
no. So they can do that,
Okay.
but they wouldn't without these extra flows caused by the defects.
Okay.
Okay? All right. So really, I only do this so I can show you the movie. All right. This was in Nature Physics about 3 years ago, this thing. And it's a small example of just how amazing nature is, but it's not a generic thing that's going to happen in all bacterial colonies by any means. Yeah. Okay, so now we better do some real work. I've talked about 2 dimensions. How are things different in 3 dimensions? And let me just tell you the punchline. The punchline is that really active turbulence is pretty much the same in 3 dimensions. Okay? The difference is that instead of having topological defects which are points, you end up with things called disclination lines. And these lines form loops. They have to form loops because else they'd shrink. And the way I like to think about it is thinking about the cross-section across the loops. The cross-section across the loops can either look like a -1/2 defect, or then this thing can twist around and you can end up with something looking like a +1/2 defect. Or in the middle, if this is my disclination line and that's my cross-section, you can also have something called a twist cross-section, where the director can point out of the 2-dimensional plane. Okay? And these things have to form closed loops, because else they would just shrink and disappear, or they have to finish on surfaces. So modulo these being different, active turbulence looks very similar. You can describe it very nicely with the same equations. You just need a bigger computer. Okay, and these are some very beautiful experiments from Nematics group, where you actually see the disclination lines in this microtubule motor mixture in 3 dimensions. The only extra thing they had to do is they had to add passive pneumatic colloids to the mixture, and that's because you need it wasn't aligned pneumatically enough without them. It helped to give you the pneumatic alignment you needed to get the active turbulence. So here are the disclination loops, and we can just watch the movie. So this is just like watching the dynamics of the topological defects in 2 dimensions. Here you can look at the dynamics of these loops. And there's a single loop, which you're just seeing now. And what you see is that these loops can appear, they can sort of pop out as little loops, and they can disappear as loops, and they can divide like that. But again, as in many of these systems, it's really hard to make this stuff. And so you can do a few experiments, but you can't use it for anything. So I agree with Aparna that the really exciting thing about 3D active matter is biology, because tissues and cells are active things in 3D. Tissues in particular are dense active matter in 3 dimensions. Okay, so that's really all I wanted to say about 3D. It's almost the same, except disclinations are replaced by disclination lines. Now I'm going to talk about this thing called active anchoring. Active anchoring is, I think, seen in biology, but again, we're at the beginning of understanding this. This is if you have a surface. So imagine if you have a sphere, say, of active material. So this is bog standard active pneumatic in here, okay? And there's some sort of surface. And so you've got something which is active inside the drop and passive outside the drop. So there's a change in the order parameter across the edge of that drop. Right, because inside you have an order parameter which says this is an active pneumatic, Q is 1, outside Q is 0, the magnitude of the order changes. So then it's the usual story, changes in magnitude of the order parameter, give me stresses, give me flows, and it turns out that what these flows do is that it tends to align the colloids or the pneumatogens or the active particles along the boundary. So in an extensile system, it tends to align them with planar anchoring. It aligns them parallel to the surface. And in a contractile system, these things tend to align perpendicular to the surface. Now, has anyone seen anything like that somewhere else? Yeah? Yeah? X-planks? Sorry, what? Oh, do they do this? Not sure the physics is the same, but might be. That's fun. Yeah? We're going to hear about that this afternoon, aren't we? Yeah, go on. That's right. I always get these the wrong way around, but I think it's pullers do this, is that right? Pullers tend to line up next to the surface. Similar physics. Pushers tend to line up like that, because the flow fields of each individual particle is really what we're talking about here. So it's a very similar sort of physics. And we've called it active anchoring because if you're a liquid crystal person, you care how the liquid crystals behave near a boundary. You talk about the anchoring of these liquid crystals, but this is just due to activity. I have no thermodynamic anchoring in here. If you just turn on the activity, the extensile ones tend to align along the surface, and the contractile ones tend to align perpendicular to the surface. Okay? This is, yeah, sorry.
So it's me.
Yeah.
In the self-propelled rod realization, extensile and contractile, would it be one in the polar axis and the other one perpendicular to it, the self-propulsion?
I'm not sure, because I think if they were pneumatic, if they were driven by pneumatic forces, I think yes, because then they'd look like swimmers. Other people know much more about polar rods than I do, so I'm not quite sure what would happen if we had polar rods. Sorry, yeah.
Thank you.
This is pneumatic physics, really. Okay?
Which kind of boundary condition?
If you have a surface, what will polar rods do? I mean, I don't think there's... Polar rods only have one direction, so you can't be extensile.
They actually tend to align with the surface.
They tend to align, yeah.
And there is accumulation.
Yeah.
It's
Yeah.
some kind of interesting
Yeah.
phenomenon.
But this is different, I think, because this is constant density. It's the direction that the pneumatic order parameter makes with the surface. This picture is just to show you that it's quite a strong effect, particularly in 3 dimensions. This is an extensile system, and this is the distribution of cos theta, where theta is the angle between the, if I've got this right, the normal to the surface and the direction of the director. So cos theta equals 0 means that angle is 90 degrees, right? So here, everything wants to align parallel to the surface. I'm in 3D, so this is a cos theta plot. That would be flat if there was no, if everything was completely isotropic at the surface, this plot will be flat, and it obviously isn't flat by quite a long way. This one here is a contractile system. Again, it's not flat, but in the other direction. So that's really quite a lot of alignment going on there. So those are 2 very basic things, 3D and this active anchoring. What I'm going to do now is show you 2 examples where they actually do things. The first is really a numerical thing, and we're trying to push it now in the direction of a bit more reality. And the second one is cell sorting. So this is due to Liam, who graduated about a year ago. And he said, right, let's take this active pneumatic model, exactly the model I was talking about yesterday, and let's put it in a droplet. All right? So I've got a drop of active pneumatic, and surrounding the drop of active pneumatic, I've got water, if you like, a passive fluid, an ordinary fluid. It's kept in a drop by surface tension here. Let's do the simulations, and let's see what comes out. This is probably the one to look at. These are these disclination lines. You can see occasionally a loop forms. They always have to end on a surface. And because this thing is active, the drop's being pushed by the activity. So it's sort of like a bag of, I don't know, it's just a bag of bacteria, really, who are pushing things around. How this thing behaves, because of the active anchoring, depends on whether you have an extensile system or a contractile system in there. And it also depends on things like all sorts of things. So I'm going to show you 3 examples of what's going on here. This is an extensile drop. And what happens in an extensile drop is that you get these sort of arms formed. So you get arms pushing out, and then they come back again. This is more active than the picture we had before, okay? So the material is pushed out, and then it collapses back again. Now, what might be pushing it out? These arms are a bit big. That's a resolution thing. Let me show you this is one where we only have an active surface, but the physics is the same. Again, you get these arms pushed out. All right, this is actually the activities just on the surface rather than the bolt. Why do you think these arms might? Anyone got any ideas why this drop suddenly pushes out arms? Have a go? Yeah? Great, well done. Yes, excellent. Yeah. These are places where you have a plus or half topological defect. Remember, the plus or half defects are self-motile, and so they will move. If they are aiming in the direction of the surface, what happens is that they push out these arms, and then they sort of escape from the end, and then the arm falls back again. That's simulations. Let's look at reality. Actually, so I want to look at this one first, and then I'll go back. Yeah.
How is it compared with the surface tension? It's the bacteria are in water.
These are simulations, so we can do what we like with the surface tension, and you have to have it low, obviously, because you're making these things. I'm going to show you some examples of reality.
And is there some chance to split the droplets?
Yes.
Okay.
Yes. I haven't got those pictures, but if you increase the activity, the droplet explodes, essentially, because these arms just escape. Yeah.
Okay.
If you had the active nematogens here, yeah.
Yeah, sorry.
If you had the active nematogens spread all over as an initial condition, spread all over the sample, do they end up forming clusters, forming this droplet after you run the simulations?
So you want
So suppose you don't start with the droplet, but you start with everybody.
In this particular setup, we have an incompressible fluid, and so we can't have that, all right? I'll show you some examples, actually, in the colloquium, where we have two fluids together, when that definitely does happen, but in a rather different context, yeah. Okay, other questions? So now let's look at some real systems. This is actually from very early paper where they looked at a vesicle of active microtubule motor mixtures. So basically you have these active microtubule motor mixtures which go to the surface of the drop. So it's like a shell of this active material. And if you look at this, you will see arms, okay? They just start when the thing gets going. You see the arms popping out? They're much thinner, and that's because we have real problems with resolution in the simulations. This is realistic, and those happen at topological defects. And I mean, this has been known way before we looked at the simulations, but nobody really noticed it. So, no, it's Hydra I wanted to say next. This was just put in the wrong order. All right. So this is just the most acute system, and the jury is very much still out on how much what's happening here is the same as what's happening in the active nematics. This is Hydra. Hydra is a little marine polyp, tiny little marine animal, millimeters in size, which sort of sits on the bottom of the sea. This is its foot. That's its mouth, through which it eats even smaller things, and these are its tentacles. What happens is if you take this thing and you cut a little square out, right? Just a little square of its edge. It's hollow, so it's sort of like its skin. Leave it there. 2 or 3 days later, it will have grown back into a proper Hydra. It's just amazing. And nobody quite knows how it does it, but what in this beautiful paper they find is that the arms grow at positions of topological defects, and indeed the tail and the mouth grow at positions of topological defects. It's not the same as the drop. What you end up with here is actin filaments, filaments going up like this, and the defect is in these actin filaments, and then the defect pattern is in these sort of long elastic filaments. So it's not really a liquid, it's much more like an active solid. And work since then, people have been trying to understand the biology. But it's hard. It's really hard. And it's a lovely example of developmental biology. So now let's go back to the simulations. I'm not going to stress this is what happens if you have a large contractile drop. Remember, the difference in a contractile drop is that the director field at the surface wants to be in a different direction, and what you end up with is these weird patterns coming out. I will explain those later to anyone who's interested. And then this one we thought was nice at the time. This is what happens if you have a contractile drop which is very small. It's a contractile drop. This is my active drop. And you can see that the director is normal to the surface. This is active anchoring. And so in the middle of the drop, remember I'm in 3D, there's going to be a hedgehog-like defect. Okay, hedgehog defect, which has all the directors pointing outwards from it. That's really bad because it's very, very high energy. So what's going to happen is that very quickly that becomes unstable. You end up with a ring round the drop where the director is in plane. The complicated director field sets up complicated flows, and what happens is that you end up with a cup shape. So just because it's active, you go from this small contractile drop to this cup shape. And the reason that that might be interesting is that in the early human embryo, indeed the early embryo of almost anything, you start with a ball of cells, and then it ends up changing into this cup shape. And there's very much the biology is much more complicated than this. But people are using these active ideas to try and understand the biology. That is very much state-of-the-art research in developmental biology at the moment. We're working on it, but that's another research seminar for another day. Okay, anyone got any questions now? We're getting...
Did you use the phase field model to buoy the droplet, or you integrated the Navier-Stokes equations?
This was these continuum equations that I was talking about yesterday. Yeah.
Perfect.
Everything I'm going to say is continuum equations till lecture 4 tomorrow. Yeah. The trouble with phase field is you can't do it in 3D. At least we can't. Yeah. Okay, other questions? Gosh, this is a lot for this time of day, but let's go with it. Let's now look at cell sorting. All right, so I'm sticking with this active anchoring, but I'm mostly back to 2 dimensions now. And there was actually a poster on this sort of thing, which is very interesting, that I saw maybe on Monday. So this is our take on this. This is an epithelial cell monolayer. All right, it's a mixture of 2 different sorts of epithelial cells plated on a Petri dish. I'll talk a little bit more about epithelial cells both this afternoon and then tomorrow, but for now they're just squishy cells which like to form layers. So it's a 2D layer of squishy cells on a Petri dish. We've got these wild type ones again. Those are purple. And then we've got ones which have been mucked around with by changing the cell-cell junctions. So 2 different sorts of cells. What they do is microphase separation, all right? So they separate into purple regions and green regions. That's the same picture with different pretty colors. Happens in 3D as well. These are actually cell spheroids where you have 2 different sorts. Now you have a whole ball of cells with 2 different sorts of cells in. And again, you get this sort of almost wetting-like behavior. You get sorting into the different colors. Now, the official story for this is just like phase separation. They call it the differential adhesion hypothesis. And basically what it's saying is that the purple cells like energetically to be close to the purple ones, and the green ones like to be close to the green ones, and so you get a surface tension between them. So just like if you have a binary fluid mixture and you cool it down, you get phase separation. Right? So that is a thermodynamic argument. It's got nothing to do with these being cells. It would work for mixture of two liquids. That might be right. But what we were thinking is that these cells are active. These are active particles, yeah? Okay? So can activity do this for you? Let's have two systems where the thermodynamics is identical. So the thermodynamics would be identical in the purple system and the green system. Will it phase separate? Well, I get this behavior if I have a different activity in the green bit and in the purple bit. And I can do that with my model, right? Because we had that zeta q thing that we went on about at great length yesterday. And if I just take zeta different for the two different sorts of cells, what will happen? So let's do it and see. I think I just have a slide for the experts. Don't worry too much about this, all right? It's a bit non-trivial to put these two different fluids together. You need a two-fluid model because locally you have to allow the amount of green and the amount of purple to change. So even if the whole thing is incompressible, locally you have to have it compressible. So you can change the concentration locally. Yeah? Yep.
So what would different activity mean at the microscopic level? Is it difference in self-propulsion? Is it difference in diffusion? Is it difference in persistence? What would it mean?
Difference in something. And I'll talk a lot about that tomorrow. We don't know. We don't know. But in the rod models, it would be difference in propulsion. That would be one possibility. Here I'm talking about difference in this pneumatic activity, but you're asking me, microscopically, what's that? I think if you took a cell out of a layer and saw how fast it moved, that's probably the closest I can get. Yeah. How efficient their machinery is of moving it around. I mean, are these things, have they got polar forces acting on them? Have they got pneumatic forces acting on them? That is still a big question. I'll talk about that tomorrow. Yeah. Okay, and you have to have these things flowing relative to each other, right? Because if you're going to have a green patch and a purple patch, you have to have the green and purple moving relative to each other so the green ones can sort of get together there and the purple ones there. So this is the technical bit. So what happens? All right, so what I'm showing you here is a movie of the concentration difference. And you can see that you get places, we started with a 50-50 mixture, and if it was passive, it would just stay a 50-50 completely mixed mixture. We turned on the activity in the yellow fluid, in one of the fluids, and then what you get is this microphase separation. It's not much of a microphase separation. The yellow bits have 0.6 of the active fluid, 60% of the active fluid, and the blue bits have 40% of the active fluid. But there's a definite phase separation there, just driven by this difference in activity. This is a picture of the same thing. This is the concentration difference as a function of time. Here it's getting going, and then it sort of fluctuates about this slightly phase-separated stuff.
Here the more active one. So what's blue and yellow again?
Sorry, the yellow bit, so it's like a phase separation. So in one phase you have more active fluid, and the other phase you have less active fluid.
So the more active here is yellow?
Yes.
Yeah.
But it doesn't really matter because it's pretty symmetric.
Well, it doesn't look symmetric.
Oh,
so
okay. Well, what happens is that the active bits are active, So what happens is the activity tends to elongate them,
Yeah, exactly.
and then they undergo the instability and break up again. So you've got this activities driving the phase separation, but then breaking up the clusters.
Yeah, so it looks like blue islands surrounded by...
Yeah,
green
I think it is the yellow's active, because you can see that the active clusters are being stretched by the activity. That's right. Yeah, that's good. Yeah. Okay. Other questions? So let me show you that you can actually get a much larger amount of phase separation. This is now, if you have a mixture of an extensile system and a contractile one. Let's do that one again. Are you going to move, movie? Okay, here you get a phase separation, which is, so it goes into almost all extensile fluid and almost all contractile fluid. But again, it's a microphase separation, because if an active cluster forms, the activity will break it up again. Thank you for being so interested.
There seems to be a wavelength there, like a pattern forming mechanism almost. Is there an instability of that sort?
We haven't managed to do a linear stability analysis. I think the length scale is just set by this balance between the time scales for things to form and for them to break up.
Okay.
Because if we add friction so that they're moving more slowly, you can sort of see it happen more slowly.
And the wavelength changes.
The wavelength changes, yes. So I haven't told you why. I haven't told you why this works, right? So this is hard. Well, it's not hard, actually, but it's this active anchoring. It's because of the active anchoring that this works. So I'm going to try and explain with this picture why the active anchoring gives you this phase separation.
Yes, but before going to the explanation of this anchoring, in the simulations that you showed before, you didn't put explicitly the anchoring.
No.
No. Okay.
No.
Yeah.
No, the two identical fluids, except for difference in activity. And the free energy looks like that, right? So there's no thermodynamic phase separation.
Okay, thank you.
So I do have to explain why they're doing this. Yeah. Okay, let's do our best. Right, so let's have a small, so this thing here is the active side, and that's the passive side. And here's the boundary between the active and the passive. And let's assume there's a tiny fluctuation, so you have more active fluid over here and more passive fluid over there. All right, so it is really an instability. So we have this little fluctuation, so this is active and that's passive. And these things are extensile, and so the active anchoring says that they're going to line up like this. So these are my liquid crystal, my active particles, all right? And they tend to line up parallel to the interface because it's an extensile system. And that's what active anchoring says. So now let me draw the flow field on there. The flow field is the red arrows, right? Because these are extensile, so they pump fluid out from the ends, and that's more or less going to balance out, and then in from the sides. There are more of them over here, because I said there was a fluctuation which made this side more active. All right? So if you look at the red arrows, what happens is that there's more flow going that way, and so if this becomes a bit active, you get this instability. It sets up flows going that way, and it becomes more active. And so it is this active anchoring which gives you this instability. And then once this side is more active, the free energy says, I don't like that, I'm trying to mix it up again. And so the free energy will tend to mix it up again. Same thing works for a contractile system because you actually have two changes of sign. Let's have a small fluctuation which says I have more of the active nematogens on this side of the interface. They're contractile, so active anchoring says that they point in this direction relative to the interface. Pop the flows on. The flows are the red arrows. Contractile systems have flows into the ends and out from the sides. Okay, so on the whole, you get a flow going this way, which again moves the active molecules to this active side. That's an instability. You get this phase separation. Now, phase separation has been seen in loads and loads of models of active systems, in rod models. If you have rods of two different speeds, you get phase separation. Maybe some of you have actually done that, and there was that nice poster on what was happening with rings as well. If I have an active system with two sorts of active particles with different speeds, they will phase separate. Okay? That's a different mechanism, and I don't understand whether it's in any way connected to this. These dry systems without the hydrodynamics do it, all right? And I don't understand why it works. I don't think anyone does, talking to people who work on that, okay? Two different speeds. If they're active, they will phase separate. This is a completely different mechanism. We don't understand if they're related. We don't understand why the two different speeds phase separate. So there's lots to be understood there.
I have a question about the microphase separation we found before. Can I explain the macrophase separation we see in cell sorting process in a lot of experiments? We see large clusters forming. So in this sense, we would need to add or combine differential activity with differential adhesion to explain cell sorting phenomenon.
I think you could do it, I mean, for example, here, if you turn down the activity as it's sorting, it would probably sort completely. All right, so that's one answer to that question. So I think by changing parameters, you could probably do it. But it would be nice not to have to adjust parameters carefully. So we did a vertex model of cell sorting. So a vertex model, all right, with plus and with active and with contractile and extensile things. And that sorts beautifully, right? That sorts completely. But it's a different mechanism. That's a combination of the rheology plus the difference in velocities. So one of my big questions at the moment is how a vertex model maps onto this sort of continuum model. We're not there yet with that. Yeah. We being everyone, I think. Yeah. Yeah. So there's a very good questions, but it's still, you know, this is real research stuff. Yeah.
No, I just wanted to comment that sometimes the difference between phase separation, microphase separation depends on how nonlinear solution gets selected. So it has to do with the basin of attraction, and it's a very delicate. It has to do with the way fluctuation act in the system, and these are a thermal fluctuation due to activity. So it's just that it's extremely difficult.
Yeah.
Often the solution are a little bit ad hoc in the details of the system. It's one maybe even non-universal thing, which depends on details.
Yeah. Yeah. Yeah.
So when you have active Brownian disks, for instance, of two different speeds, I believe they phase separate because, I mean, in a region rich of red particles and a region red of blue particles, meaning different speeds, basically meaning because they have different tendencies to form motility-induced phase separation, right? So locally, there is a short-range attraction between the particles due to activity, but this tendency is different. So it's kind of
Actually, yeah, so you're saying it's MIPS done differently in two different systems.
Yes.
That might make sense, yeah. That might make sense. But then it still works if you have systems which are not MIPS forming.
Yes. If you have a dense system, you still have these separation, segregation in this sense, or sorting.
Mm-hmm. But I think if you just have ideal gases, two mixtures of ideal gases, I think, I might be wrong, two different active ideal gases, those will also phase separate. I don't think, but we can, anyway, we can discuss. I mean, I'm certainly no expert on that, and I'm happy to stand corrected on that one. Yeah. Okay, so let me just finish this, because this is another of these cute things like Hydra. This is a starfish embryo. You have two different sorts of cells which have fancy biological names, which are well translated by a physicist into red and green. Makes it easy, like your underground system. Okay, and basically you put these in a blender, and then you get loads of red and green cells like that. If you just leave them there, they will undergo microphase separation, and then they will organize themselves into this proper starfish embryo again. Somehow these things, these cells, are able to get from there to there with no help whatsoever. I think that's absolutely amazing, and I don't understand it. It's a good problem for someone. And that's just a starfish. I mean, we have to do the same, right? We start as a tiny ball of cells. We have to end up with us. So there's a lot to understand there. Good. Has anyone got any particular questions there? Because we're going to move on now. We've done that one. We've done that one. Any questions? Right. Okay. So what comes next is confinement. I've talked about active turbulence, but if you're a biological system, you could probably do without active turbulence. Certainly in terms of the tissues in our body, we could do without active turbulence, right? Because, you know, that's not really a biological thing. It's just a waste of energy having a chaotic system. And therefore, maybe confinement makes a difference. So what happens if I take my active nematic and I put it in a channel so it's confined? And the answer is that you get a series of phases. First of all, you get no flow at all. And then if you increase the activity or the width of the channel, you get a laminar flow, i.e. a flow which just flows in straight lines down the channel or is maybe a shear flow. And then you get a fun thing which we call the dancing state, for reasons I'll show you, or a vortex state. And then you get active turbulence.
Julia, does it matter what the anchoring is, if it's homeotropic or
Sorry,
planar? Does it matter what the anchoring is, if it's homeotropic or if it's planar? Or?
No.
Okay.
Two comments on that. It's normally homeotropic because that's what these large colloids do at a surface. The anchoring will make small differences, but not qualitative differences. Yeah. Okay. So the control parameter for this is increasing activity, or this is also increasing channel width. Okay, so why do you think you might end up killing the active turbulence? What's not going to fit into the channel? What's not going to fit into the channel? The vortices, that's right, yeah. So really, what's happening is you can't even get half a vortex in here, half a vortex sort of gets in and you get the laminar flow. This is actually one vortex across the channel, and then this is active turbulence. So it's because you can't fit the instability into the channel, or you can't, saying the same thing, you can't fit a vortex into the channel. So I'm going to just give you mostly pictures, but starting from the no-flow state and sort of moving through this to see what these things look like. There's a really, really beautiful paper, this one, where they solve the hydrodynamic equations of motion that I showed you for one of these things in a channel, and they actually show the instability which gives you the flow, this laminar flow. It's the same instability it's the same instability that gives you the active turbulence, but now it's confined in a channel. So what it does instead is that near the crossover, you get a directive field like this. Because you get a directive field like that, here you have large gradients equals stresses equals forces, and so they're pulling the fluid like that, and you end up with a profile which looks a bit Poussin-like, but it tends to be a bit flatter here, so it's being pulled like that. I really like this. It's a lovely paper. It would take a lecture or more to go through it, but it's another way of looking at this active instability. So these are experiments. These are experiments in Francesc Sagre's group. And what they did is they wanted to look at this flow in channels. And so they put this active microtubule motor mixture here. This is a two-dimensional layer. And then they plonked this, what's it called, this thing on top of it. Cover slip? I don't know. This thing with holes in, anyway. This, and by using this, they'd have microtubule motor mixture underneath it, so they'd have the same stuff in each of the channels, which makes it easier to compare them. And the sort of width is, these are 30 microns, and these are about 400 microns. So let's have a look at the results of the experiments. Right, so this is laminar flow. And because I've got ends to my channel, I can't just have that sort of laminar flow. So what you get is shear flow, goes in this direction at the top, sort of turns around at the end and comes back at the bottom. And you get shear flow. So this is a nice laminar flow seen in experiments. But then you suddenly get these bursts of topological defects occurring. All right, this wasn't originally predicted in the theories, but we do now understand them. What's happening is that the shear flow is pulling the microtubules so they're nematically aligned. And the minute you have things which are nematically aligned, they become unstable and they bend like this. And that's like a load of topological defects appearing. Let me just put that again. You start, behave. What's my done? I turned it off. Maybe the battery's gone. Okay, so you start off with simple shear flow. Microtubules get pulled out, and then you see the instabilities which look like these bursts of defects. These are pictures of this in different geometries and different systems. This one's exactly the same thing, but in a circle. So the laminar flow now goes round in a circle, and then you suddenly get one of these defects appearing, and you end up with this rather lovely picture. Parna might be a co-author on this. I'm not sure where. Okay. And we'll see exactly the same sort of thing in a confluent cell layer. These confluent cell layers also take off and start going round in circles, which is another reason for calling them active nematics. And I've got my favorite video on this, but I'm not going to show you till the talk this afternoon. Yeah. Okay, so that's sort of here. Now let's make the channel a bit wider so we can actually fit the vortices in. And then you get this most amazing structure where you get a line of vortices. So what I'm showing at the top is a line of vortices, but they're dynamical vortices, and you get topological defects which go in this direction around the vortices, and at the same time topological defects which go in that direction along the vortices. Here's probably the nicest picture of that. Those little green things are plus or minus, sorry, plus or half defects. The minus ones go and sit at the walls. These are simulations. In a real system, life is not that nice because you have fluctuations and goodness knows what happens. But in Frances's experiments, he sees something very similar, but the defects are just less long-lived. The fact we saw that lovely pattern in simulations was due to Chris Tyson being very careful with boundary conditions and initial conditions. If you just sort of throw the stuff in without worrying, you get something of this pattern, but it's much more random. I'm not going to talk about that. Right. So confinement. You get all these different states, and maybe in real life, well, certainly it's known that cancer spheroids, where you have a three-dimensional cancer spheroid, starts going round. Experiments have shown that, which is, again, another reason for thinking these might be active. So I'm going to finish by I'm not going to do this because we can do it tomorrow instead, but I'm going to give you the question, right? We've talked about things being active nematics, and for the microtubule motor mixture, they're the right shape. Although if we turn off the activity, what this microtubule motor mixture does is it doesn't go back to a very nice nematic phase. It sort of plumps at the bottom of the system. Similarly with bacteria, right? If you turn off the activity, they just sort of, they don't form a nice regular nematic. They form something which I guess you could almost call an isotropic phase with everything random. So instead of starting with a nematic, if I started with an isotropic phase, can I still get active turbulence? That's going to be very much closer to the experiments. And to make it even worse, and I will say something about this afternoon. I hope I'm not going to get mixed up and go in the right direction in these talks. All right. The other thing is, I mean, you were telling me the other day with cells. Cells start circular, right? So they're not even, those are certainly not nematic. How can these circular cells end up giving you active turbulence? And in both cases, the answer is yes, they can, because there's another instability which makes them nematic, and then the nematic instability destroys the nematic order, and there's other instability makes them a nematic again. But I think that's a story for this afternoon and tomorrow. So I'll stop there. Thanks very much.
Questions?
Okay,
I do have a question.
Okay, but they've asked lots of questions, so they've been doing well for questions, yeah.
In all these interactions that appeared, they were local. You had long-range correlations, but no long-range interactions. But
the hydrodynamics is long-range. Hydrodynamics is by its very nature long-range, so we really are on the continuum here, right? So we're thinking of it in terms of molecular interactions. That's all been washed out. But yeah, momentum transfer is long range. So yeah, we do have long range interactions.
Okay. No, I think in the case of Hydra.
In Hydra, yeah.
In the regeneration, you, besides the movement of cells and the inflow of water and outflow, you also have a neural network. And so this would make a long-range interaction for the cells, not just local.
Hydra is probably as much elastic as fluid, or indeed more elastic than fluid. I mean, the +1/2 defect idea is okay, but I think the details are very different from Hydra.
Okay.
It's biology, which is not really understood. Yeah, that's right. Yeah.
I have a question about the active anchoring part. You have a difference, for instance, in the shapes when you had extensile versus contractile. And my question is a little bit vague, but I'm wondering whether people have explored the differential geometry of the surfaces depending on this, for instance, looked into the Gaussian curvature, et cetera, and whether there's anything interesting coming out of this.
I think in 3D, no, because it's a mess.
Okay.
You could do it, but I suspect it's so far away from experiment that it's hard. In 2D, you definitely see if you have a curved surface, the defects. Do I mean that? No, I mean a surface in 3D, so that if you have a place with negative curvature, +1/2 defects like to go there, and a place with positive curvature, the -1/2 likes to go there, except I might have got those the wrong way around. But defects like bits of curvature because then that reduces their elastic energy. And there's been quite a lot of work on that.
Okay, got it.
Thank you for the lecture. Starting for the experiments with the both different types of activity, could I reduce the experiment to an activity particles, like bacterial rod-like, with passive rod-like, and expect that the passive ones will get, pushed or pulled by the another ones, and dislocate more, or the passive nature of this mixture will annihilate the active fluxes induced?
No, in this hydrodynamic theory, the first one I showed you was active-passive, so it works there. If I have active-passive bacteria, I think it would work, but I'm not certain. Because bacteria tend to crawl on the surface, so it's a dry system. That's why I might think it would be different. So yeah, I'm not sure, actually. That's interesting. So live and dead bacteria, would the dead ones cluster, driven by the live ones? Yeah, I suspect someone's done it, but I don't know the answer.
Okay, thank you.
Is there any kind of theory that accounts for or is based on the kind of anchoring you have being developed or already developed that you're familiar?
I mean, what I said is based on, we know how to write down the forces on the surface, and therefore we know how to write down the flows on the surface. So yes. I mean, I think yes is the answer. Yeah, I'm just thinking if we need to add more to that. But yes, the picture is underlined by theory, because that zeta q thing, you can work out what it looks like near a surface.
Oh, yeah, just to complement about Danilo's question, Rodrigo's group here has a channel which has a surface with a sign-like structure. They put E. coli there, right? You can put more particles and make them do nematic stuff there. Could be interesting.
Yeah, I mean, there's quite a lot of work by Ray Goldstein a while ago of bacteria in a channel. But not with a surface like that, no.
I just have a comment on the dead bacteria. I don't know if it has been done, but in principle, they should enforce the same mechanism of slowing down. I mean, if you collide with a dead bacteria, you slow down, and this is the feedback mechanism of MIPS. So in principle, it should work. Even if then you may have different center of aggregation, which does, so you may have an arrested MIPS, something like that. I don't know if there are experiments, but principle.
I bet there are, but that makes a lot of sense. It's like having a surface, really, isn't it? Because the dead bacteria are sort of being a barrier. Yeah.
No more questions? So we thank our speaker again.